Okay, this is section 5. This is section 5 within chapter 5. The topic in this section is active transport. So, active transport is a movement of material up or against a concentration gradient. Basically, we're pumping material uphill. We're pumping material from where it's in low concentration to where it is already in high concentration. And in order to do that, cells have to use energy from the ATP, adenosine triphosphate. And so movement uphill does not just happen on its own. Now, an example of active transport, again, movement of materials to areas where they're already in high concentration, is the sodium-potassium pump. And the sodium-potassium pump plays a number of different roles in the human body. It's associated with the function of neurons. It's associated with the uptake of glucose from the digestive tract, along with a lot of other functions. So, this sodium-potassium pump, it depends on the establishment of what's called electrochemical gradients. And this electrochemical gradient is a difference in electrical charges across a membrane, as well as a difference in ion or chemical concentration across a gradient, or across a membrane. Okay? So, in order for a neuron to conduct an electrical impulse, it has to maintain this electrochemical gradient. And it's a sodium-potassium pump that establishes and maintains this electrochemical gradient. So the sodium-potassium pump is a carrier protein, which means it's going to attach to the sodium and the potassium, change its shape, drop the ions off on the other side of the membrane, return to its original shape. And in this case, a carrier protein can be involved in facilitated diffusion, or it can be involved in active transport. In this case, it is active transport. The difference is, in facilitated diffusion, the material is moving down a concentration gradient, down or along a concentration gradient. It's moving to an area where it's in lower concentration, and the cell does not utilize any of its energy to cause that to happen. In active transport, the material is being moved up a concentration gradient or against a concentration gradient, and the cell has to expend some of its ATP energy to cause this to happen. So, this movement of the sodium and potassium creates an electrical difference and a chemical difference across the membrane. So you notice right here, I'll show what's happening is 3 sodiums are being moved out, and in return 2 potassiums are being moved into the cell, and ATP is breaking down to ADP, adenosine diphosphate, and inorganic phosphate. And in the process, that's using some of the energy from the cell. Okay, so see if we can go through this diagram right here as to what's going on. And we'll start right here. And you'll notice that we've got 1, 2, 3 sodium ions inside the cell, and they're going to fit into these receptors in the protein, and then we'll have phosphate from the ATP attached to this protein. So we're going to phosphorylate the protein. Well, that causes a change in shape, which dumps these 3 sodiums to the outside of the cell. Now then, this protein can pick up 2 potassiums from outside. They attach to the protein, causing the protein to change its shape. It releases the phosphate, also involved in changing the shape of the protein, and the potassiums are dropped off to the inside of the cell. So, as this process goes along, here's a basically right here's a neuron. Okay, so there's a neuron, and we are going to use ATP energy from the cell. So ATP is going to be the source of energy to drive this. We're going to pump 3 sodium ions out here. We're going to turn around and pump 2 potassium ions to the inside of this neuron. So you notice we have a difference in electrical charges. We have 3 positive charges on the outside of the cell, only 2 positive charges on the inside of the cell. So we've got more positives on the outside, fewer positives on the inside. So we regard this as being positive charge on the outside, negative on the inside, but that only means that we have more positives on the outside than on the inside. We also have a chemical difference. We've got an abundance of sodiums now on the outside, an abundance of potassiums on the inside. So these neurons expend a lot of energy in the form of ATP to maintain this gradient. We maintain that gradient so that if we have some form of stimulus that activates this neuron, that stimulus could be touch, it could be sound, it could be sight, some kind of sight, light, some kind of stimulus activates this neuron, it is going to open up gated channel proteins that will now allow the sodium ions to come pouring in. So the sodium ions come pouring into the cell, and potassium ions go pouring out right here. And I'm about out of space there to run that, so they go pouring out, and that's depolarization. And that depolarization will propagate itself along the neuron, and that way convey an electrical charge along that neuron. Now, as soon as this occurs, this sodium-potassium pump goes back to work using energy to reestablish that gradient of sodium ions and potassium ions and that gradient of charges. Likewise, just to maintain it, we expend a lot of energy just to maintain that gradient in a state of readiness. Okay, so there's our sodium-potassium pump. Let's go on over here. Well, wait a second. Let's go here. So there we go. Another form of active transport is one called co-transport. This is where 2 molecules are transported together. And one molecule is transported against its concentration gradient, in other words, uphill. The other molecule is moved along its concentration gradient, essentially downhill. And so ions like sodium, potassium, hydrogen will move along their concentration gradient. The second molecule goes along with the flow, even though it's moving against its concentration gradient. It's moving uphill. So let's go back here for a second, and let's look at a diagram for that. So we have a cell. Whoops. Try this right here. So right here. So we have a cell right here. And out here we have an abundance of sodium and glucose. Okay? So we have a protein right here that can move these two together. So we have an abundance of sodium out here, we have less sodium in here. We have an abundance of glucose in the cell already. So, what will happen is the sodium will move through. The sodium is essentially going downhill. It's going along its concentration gradient, and glucose just kind of hitches a ride with this, even though the glucose is going against its diffusion gradient here. And that process is called two things together, transport. Now, co-transport is indirectly a form of active transport, because somewhere over here we have to have energy being used in the form of adenosine triphosphate to run this pump, to keep pumping the sodium, let me find its route through here, keep pumping the sodium back to where the sodium is already in high concentration. So we have to have a pump to maintain that concentration gradient so the sodium can keep flowing in there to carry the glucose along with it. So co-transport indirectly is a form of active transport. Okay, so, whoops, let's try this. There we go. So, co-transport indirectly requires ATP energy. Thus, co-transport is active transport. Co-transport itself is passive, but we're going to have to use ATP energy in order to establish that ion gradient. Okay, so here's the example, and you notice you have the abundance of sodium out there in the outside the cell. You have a lesser amount of glucose. So, right here, the sodium is going where it's in high concentration to where it's in low concentration. The glucose is going to where it's already in higher concentration. So the glucose just going along for the ride. Also, going back to, we talked about carrier proteins and channel proteins. Channel proteins are the ones that just act like an open pipe. Carrier proteins are the ones that bond with the material that they're transporting, change their shape, and carry the substance across the membrane. So, the co-transport itself is not an active process, but somewhere in the system we have to have energy being used to maintain this abundance of sodium ions out here on the outside of the cell. Okay, so co-transport, examples of that. Well, we've got glucose and sodium co-transported from the digestive tract into the epithelial cells lining the small intestines. We've got the fact that sodium is moving along its concentration gradient. Glucose moves against its concentration gradient. Then the glucose will in turn be moved by facilitated diffusion from where it's in high concentration in the epithelium to where it's in a lower concentration in the blood. And then the epithelium has a higher concentration. It's a high concentration area. The blood's a low concentration area. And the concentration of sodium outside the epithelium is maintained by sodium-potassium pump, utilizing energy from the cell to maintain that high amount of sodium for the epithelium, so the high amount of sodium outside the epithelial cells. Okay. Now we've got another item right here that's not really discussed in your textbook to any great extent, but it's kind of an interesting topic, and it does show up on your test. And so this is called the patch clamp technique. And this is a technique that allows the study of individual ion transport proteins.